Hello friends, welcome to the first lecture of week 4. In the past lectures, in the last week, we have looked at Linux, how to run the Linux commands, and also how to use R for various genomic analysis, right? And now in this lecture, we will look at the available programs which are publicly available for usage, right? So you can use it online as well as you can also download most of them, and also by local installation you can use them at your terminal, okay, if you have the sufficient capability of the system and like that, okay. So both have their own advantages and disadvantages, and then we will also look at the some overview of what is high performance computing and what kind of systems are generally used. So there are several publicly available packages, websites, resources which allow you to do a lot of daily routine genomic analysis online, okay. And if you when you become little more familiar with this, and okay, by little searches, reading journals, articles, you will know that basically academic institutes, groups, researchers are releasing a lot of developing a lot of software which they make a GUI based application or a downloadable application which you can download from internet and use it, okay. For example, GitHub may have a lot of scripts and programs that you may like to use, right? So, and also like when you do Perl, Python, or even R scripts or Linux scripts, there is always a possibility that somebody else have developed the same and you can find it online and then or partially modify it and use for your own purpose, okay. But today basically we'll look at the resources which are standard and available online, and which should be very helpful for your routine analysis, okay. So for example, like EBI site offers a lot of resources for common genomic analysis, structure predictions, right? EMBOSS is one of the tools, a suite of packages that is available. NCBI offers ORF Finder and Clustal W by EBI again. Okay, so EMBOSS is European Molecular Biology open software suite, which is a very commonly used suite of softwares, okay, or basically a consortium of software, group of softwares or tools which allows you to do routine analysis, as I mentioned, okay. And basically it has hundreds of programs, applications of various types, and you can also download it. So like for example, sequence alignment, if you have like smaller tasks, you can always do using this. Searching something in a sequence, finding a motif in a sequence, right? Predicting ORFs in a sequence, translating a sequence like from DNA to protein, or back translate okay, from DNA to DNA, or basically reverse complement a DNA sequence. All these things for which you may think of writing a program is already available and you can use it directly. Call it in your Perl script and you can use it. Okay, so basically it's a very robust bioinformatics application tools, right? And there are so many tools that it is probability that like all the commonly available you should be able to find in this package, okay. And basically it is easy to use, open source, free of charge, right? And that is why like you don't have to subscribe to it or pay for it or buy it, okay. Available generally for like major platforms, OS, Windows, Linux or Mac, or okay. And most of the applications have a similar interface in which you select a program, upload a sequence, change some parameters if you want, otherwise the default ones are always good, right? Otherwise you can change according to your need, and right, and then submit and you get a result. So we'll go through this, okay. And basically it does not have any like limitations as such that you have to submit only this size sequence or this much is allowed, okay. So basically it is free for such kind of limitation. Okay, so many things you can do: sequence alignment, finding motifs, finding ORFs, right? Finding CPG islands, repeats, okay, codon usage, okay, peptide statistics. All these things are readily available here, and it's just a matter of that I will also show you few of those with examples, right? What was the program like, what was the input, and what was the outcome of that, right? and then you can go to the website, and which I'll suggest, and you can check for the remaining options and do this on your own. Okay, so this is the EMBOSS website to use it online. So it is offered by EBI, ebi.ac.uk is the site. Okay, EBI AC is Academic Institute and UK is for United Kingdom, right? And it has a suite of EMBOSS related software like EMBOSS Back, Trana Big, okay, back translated protein sequence to nucleotide, okay, back transseq, okay, translated protein sequence to nucleotide using a codon frequency, right? That what kind of genome is that, based on that, okay. And similarly there are so many tools which you can find on this page, okay. And so basically EMBOSS has a web interface, okay, as well as a command line option. So web interface first we will see, and then we will go to the command line option that you can run on your local system. Okay, so this is a web interface. You can access it using this link. Okay, so list of programs is now appearing here, and then you can, what you can do is you can select. So basically a brief description of the program is given, like cons will create a consensus sequence alignment from a protein or nucleotide, that what is common between them, it will just highlight those and light, okay. So like a consensus from those, right. Similarly needle is there for local the global alignment, sorry, for two sequences, right. And needle is a standard algorithm which also is used for like other further developed algorithms. Okay, so okay, so basically these are the two, cons and needle. Right, now what kind of sequences you can enter or it can accept. So generally all the good or widely used formats like FASTA, which we see in the last class, FASTQ, that is the typical Illumina format with with the sequence quality scores. Genomic format, PDB, Clustal, EMBOSS, okay, plain text format also, and PHYLIP. So these are the formats which are standard outcome of various programs or commonly used in genomics, and all these formats are acceptable by EMBOSS. Okay, so now I'll be using these sequences. So this is a protein sequence, okay, with a in FASTA format with a header. This is the name of the protein, right, annotation, and the sequence, right. And there are a couple of nucleotide sequences, test 1, test 2, test 3, right, in the again in the FASTA format, right, annotation and sequence, annotation, sequence, like that. And all of and I'll be using these sequences later to run the various applications. Okay, so like for example running a multiple sequence alignment to find a consensus, right. So like cons will be consensus, cons will be the tool. So the sequences that I just showed, test 1, test 2, test 3 are here. So either you can paste those sequences there, okay. So basically this is a GUI, it's a simple format. You have to give your input here. So you can select between protein or DNA, okay. Then for example right now we are dealing with DNA. So either I can copy the sequences and paste here, or I can upload from here, okay. Choose file and it will upload the sequence from your system, right. And also, or if you don't have a sequence, you just want to see that how it will work, you can use an example sequence. So generally these kind of tools always have an example sequence, so that you can run on a model sequence, know the outcome, and then try your sequence, okay. Then there are some more parameters that you can always change, right. We'll not go into details of that, but more knowledge of what you are going to do, and basically how you want to results will be fine-tuned, can be done using more options, okay. You can always define a title for your job, like task 1 or date wise, for example job 1 underscore the today's date, like that, okay, and then say submit, okay. So most of the tools that will be are available will have the typical this kind of format. When you submit, it will go into a queue, or it can directly be processed. If it is very big, it may go into a queue, otherwise you will get the results directly, okay. So basically your job is now queued, and you will be getting the results, okay. So it's a standard line that they paste, right. and basically they give a job ID for you, that your job with the input sequences that you have given, the program that you have chosen, okay. So that is available assigned to this link, and basically you can always copy this link, okay. So now for example you have given a small job or smaller set of sequences or small task, it should be completed within less than a minute or 2 minutes maximum, right. But if it is a very big task, then and there are chances that your browser window will close or something may happen, okay. So you can copy this link and paste this link after some time in the basically the IP address window of the any Google Chrome or Mozilla, right, and it will fetch the results for you, right. And basically this will also be saved for some time, which you have to check that how much time your jobs will be saved, okay. And then this is the job output, and okay, so basically for 3 sequences that I gave, it made a consensus of those here, okay, which was common between them, they are here, otherwise it is n, right. And this is the job output for the sequence, okay. You can download it or copy paste it, or keep the link safe for later view also, right. So this is like the online application, but many times you may like to do things locally, because it is more convenient to call these kind of small programs in your Perl script or Linux script, right. So that you can have like 4 or 5 programs together, okay, queued one by one, so that you can run all set of programs on a single file, okay. That is a routine task, for example, in sequence analysis. So you can make a script out of all those programs by calling them one by one. So basically that in that case, downloading EMBOSS, and if you are like typical genomics analysis person, then EMBOSS will be a good tool to have in your locally installed in your PC, okay. So you can download it from this site, okay, and this is the file which is star.gz. We have seen like these kind of files earlier classes, and you have to untar it and unzip it, okay. So tar minus zxvf, there is always a space here, okay, zxvf, and basically it will be uncompressed to your folder, okay. So okay, either 2 steps, like you can unzip first and then untar, okay, or you can do together like tar minus zxvf, and basically it will untar and unzip together, right. So then it is now available in your folder, okay. Now where to store these kind of programs, right. So it is always good to be organized when you use Linux, right, otherwise it will be all cluttered, and even you will know that what is the correct file, where is the file, and okay. So you should always make a program, a folder called downloads, a folder called software, right, and a work directory maybe for your work, right, or based on the project title, right. So in downloads you should download the things, right, and then after the download is done, you can move it to the software folder, or where if it is software, or if it's a database file, then you can move it to a database folder, so that things are organized in your system, okay. So now we have downloaded in downloads, and then we moved it into software folder, right, and then we have to install it, okay. So basically /configure, we have seen earlier also, this is the command prompt, okay. At the command prompt, write /configure, so it will check the configuration of your system and create a make file, okay, for your system, right. And that make file is basically accessed by this command make, so it will use that make file to install the binaries, okay. And then okay, so basically then you are ready to go, okay. But sometime like it could be that you already have a pre-version existing, and you want to install a new one, and you don't want any kind of intermixing of libraries, or okay, and you clean the previous one and install a new one, and basically you should do this way. So basically this command, remove config.cache, okay, make clean. So it will remove all the previous binaries and their paths and everything. Okay, then again run .configure for the new installation and do make. Okay, so this part will clean the previous files existence from your system, and this will install a new version of that, right? Okay, conda can also be used to do this, like we have seen earlier, conda install -c bioconda and emboss. So this is the standard command, okay, and the program name. So if it is available in the conda directory, it will fetch it, okay, and install it on your system using all the variables, settings and libraries that are needed for to run that program. Okay, another way to do this is this way. If the previous command does not work, but generally this is good enough. But in case of troubleshooting, if this is not working, then you can try this one. Okay, now how to see that what kind of programs are available in your local package and what do they do. So there is a command called was name. Okay, so this will and you can give the program name, like anything that can help me in sequence alignment. Okay, we don't know the program, right? So it will, if you give this search word, okay, even like was name alignment, if I give, so what it will do, it will find all the programs in which that option is available. Okay, like this is one cons which can help you in sequence alignment, right? And anything that has related to sequence and alignment thing will be displayed on your system. Then you can select that the best one from these, that what kind of alignment task you are going to do. Okay, then there are like several good tools like water, needle, stature for global and local alignment, and basically to know patterns in a protein, fuzz nucleotide, translate and pro, right? Annotations like TF scan or okay, like that, identifying the transcript factor binding sites in a given sequence, right? For motif discovery, okay, program for antigenic or motif, right? PatMatLab, DB. So there are several programs. I have just written a few of those, but if you search the EMBOSS directory, you will find so many programs that could be of use to you. Okay, like sequence secret for converting between different sequence formats, right? For translating reverse sequence, okay, 6-pack and reverse sequence. 6-pack will translate a sequence into 6 reading frames, okay, because DNA has 6 reading frames as we discussed earlier, okay, and reverse seq can reverse complement a reverse a sequence, and reverse refcom can reverse complement a sequence, right? And splitting a sequence into smaller sequence, okay, many times this is needed. Okay, so basically these are the routine things in genomics, and those kind of programs are already available at your disposal using EMBOSS. Okay, ORI prediction, codon usage calculations, finding the nucleic acid properties like GC. This is one of the useful programs. Like you have a nucleotide sequence, you found a new genome, you want to calculate the GC content. So just say GC and the genome file name. Okay, it will tell you what is the GC content, composition of GC, all 80 GCs in that, right? Okay, and also like for plotting also. So basically these are very useful programs for the basic analysis. Okay, finding properties of a protein, PEP-stats. So like you are developing a AI based tool or ML based tool, you want to identify all the statistics of a protein, right? So basically PEP-stats can directly give you those statistics, and you don't have to write a program on your own, because if you write it, okay, then chances are that you will spend a lot of time in optimizing and correcting for errors, and okay, but here you have already standardized and programs which are basically standard as well as ready to go. So you don't have to spend your time and energy to do like these things which are already there, but use that time for improving your main algorithm that you are developing, right? So similarly amino acid properties or nucleic acid properties, right? So hydrophobic movement and so many things are there, primer finding, okay, designing primer. So these are all the routine tasks. Even like this is very useful for experimental people, that they found a primer, they want to search that how the primer will perform, or finding a new primer for a sequence. So all these things are here. Okay, so now we will do some demonstration of the command line, how to use it command line way. Okay, so these are two sequences, sequence 1 dot FASTA, this is the file name, okay, and this is the content of that test1. Similarly, this is the file name and this is the content of that, okay, and this is another sequence, right. This is the file name protein sequence 1 and this is the content in that, because file name may be different than the actual header of that sequence or annotation of that sequence, okay. And one more sequence, protein sequence 2. So we will be using this in the further analysis. So now you want to do alignment of two sequences. So basically you are taking sequence 1 dot FASTA and sequence 2 dot FASTA. So the program is called water, it will do the alignment. So water, okay, sequence name, A sequence and B sequence, right. So this is the first sequence, this is the second sequence, A and B, and out file name, okay, which is the alignment.out. So basically it uses these kind of prefixes before a submission a file, so that you will always know, okay, that what is the file that you're talking about. For example, if I do -b sequence 2 dot FASTA here, okay, this place, or I can move it this place, or even out I can define first, but as so long as I am writing this, like out file is this, okay, wherever it is placed in that line or in the command, it will always be the out file which is named this, right. So the order will not matter there, because you are already defining a qualifier or the tag for that particular file, okay. So water A sequence, B sequence, out file, and this. So it will do the Smith-Waterman, that is the local alignment of these two sequences. We will be covering this Smith-Waterman and Needleman-Wunsch algorithm later, okay. And using the default gap opening and gap extension penalty, it will align the sequence and give you the summary that, okay, the identity between the two sequences this much, these many base pairs aligned, okay. This is the similarity, okay, which is same as identity in this case, otherwise similarity may be little higher also, okay. And the gaps, similarity may be little higher in case of proteins, but in DNA it should be same as the identity, okay. And gaps means like how many places there was some nucleotide missing at the corresponding okay, position in the two sequences, and total score is 62, okay. So basically okay, so and the output file for this, the previous one is already saved in a system, which you can use it for further analysis, right. Similarly, like converting a sequence format from one to another, okay. So I want to convert the FASTA format into maybe EMBOSS format, okay. So it will change that, okay. So secret sequence is this and out sequence is EMBOSS format, and this is the output sequence, right. So it will make this file where it has converted the sequence 1 dot FASTA, which contained was of just one sequence called test1, if you remember, and you can go back and check, right. And it will convert that into the EMBOSS format, which is this, okay, which have ID and these kind of qualifiers, sequence, SQ and // right, and some description, whatever was known from the header or annotation, it has put here, right. So converting these sequence formats could be very easily, you can see like just one line you write and it converts the sequence format, right. And basically it will be very important and needed for several applications where they may demand this kind of format, but you have the other one, and this tool can help you to convert between the two. Okay, finding statistics of a protein, properties such as molecular weight, okay, or GC content of a sequence, right. So we have tool like PepStats, okay. So basically again, like simple command line is, so basically at the $ prompt you will be writing PepStats, okay. Sequence name minus sequence is this, output file which is this, right. So basically it will give you the statistics of that. So the sequence that was inside this file had a header this MGYG and this, right. So basically the sequence was greater than and this sequence, right. Annotation line contained this header, right, in this file, okay. It had 1 to 877 characters, Okay, so it found the molecular weight, average nucleotide, average residue weight, isoelectric point, okay, charge and other statistics for the that protein. Okay, and details are here, and like more statistics, and then what are the different type of residues, like small ones or very small ones, aliphatic, aromatic, non-polar, polar amino acids, charged, right? So all these, what is their respective number and their proportion in that is mentioned now in the statistics. So basically just one command gave you the statistics for the entire protein and also for the multiple proteins that may be present in that multi-FASTA file. Okay, now another example to look for ORFs, or ORFs are basically potential genes, okay, which could be assigned called a gene if you find a function for that, or in subsequent steps you confirm that it's the ORF is really a gene, right? Otherwise it is open reading frame. Okay, DNA has like 6 reading frames, of which some could be open reading frame which are long enough, okay, and have a start and end codon, and in between there are no stop codons but valid codons of a particular distance, right? So this is like, for example, one part of one genome. So dot 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 here means that it continues further. So it's a long file. It's a Mycobacterium tuberculosis S37RV genome, and we want to find ORFs in this. Okay, so basically the program is to find ORF is get ORF. So get ORF sequence is this, right? And then this is the basically the file inside that sequence tag that we used. Okay, and this is the input file name. Okay, minus find one, okay, and outseek. So basically this is, you want to find complete ORFs, incomplete ORFs, partial ORFs, right? So basically find may have such kind of option. So it's an additional qualifier that you have added, okay, and the output sequence file. So okay, so this is the, we have written in the command prompt, okay, and it is finding and extracting ORFs and saving them in this file, right, which looks like this. So this is the output file that was made in the previous program, and you can see that for this sequence, right, this is one ORF from 8 to A118, and in this file another ORF is so ORF number 1, ORF number 2, 3, 4, 5, 6, okay, multipolar it has found, and starting from this location to this location, this 401 to 493, 671. So this, at these locations there are potential ORFs in, okay, this genome, right? This was the annotation. And reverse sense is like they are present on the complementary strand, on the crick strand or negative strand. Okay, so forward strand also have ORFs and reverse strand also have valid ORFs. Okay, and now this is a Mycobacterium tuberculosis protein sequence. Okay, like this is one sequence, this is another sequence, right, and so on. There are several sequences in this file, right, and then you will be using this file to say find some pattern, okay, or motif, okay, which is by the tool say FuzzPro. So again same syntax, program name, pattern you are defining, it could be ALT. So ALT means either it is alanine or leucine or threonine. So when you write a motif in this way, okay, ALT, so either of these could be present, either A or L or T amino acid in that sequence. Okay, so in motif I can tell you little more. So if I say XXX, right, so that and T and say G, okay, so that means that T has to be the first amino acid, then there could be any 3 amino acids XXX, then in square bracket any one of these 3, if I can write 3 or more or 4 or 5, anything you can write, okay, based on the real situation, the motif situation, okay, what the is actually found in the motif. So any one of these, and the last amino acid is G. So you can make such kind of motifs and find, okay, and it will search in your sequence and give you wherever this is occurring in the sequence. So it was just single amino acid you are finding, so it is occurring from start to end location it is mentioning, If it is a long motif, it will tell you like longer length also, 2 to 5 or 2 to 10 like that. But here it was just one amino acid, so it is just mentioning one amino acid. Okay. Right. Similarly, antigenic motif is another option that you can try and look for the antigenic regions in a protein, okay, which sometimes is important for calculating the antigenicity of a protein, that for various research purposes. Okay. Similarly, like translation and reverse complement of a sequence, like I can do transseq translation of this sequence 1 into protein. So I am translating a nucleotide file into a protein, right? So this is the sequence, right, and it is translated into this. Okay, so ATG becomes M, okay, AGT becomes S, right, CTC become L, so like that. 333 codons basis, it has translated that into this amino acid. Okay. Now similarly, like this was my sequence test 1, I can do reverse complement of this just to know that what is present on the other strand. Okay. So basically this is the forward strand 5 prime to 3 prime, and 3 prime to 5 prime is the reverse strand. So if this is the sequence at the forward strand, okay, I can find the sequence for the reverse strand by using reverse seq. Okay, so this will do a reverse complement, reverse and complement of a nucleotide sequence. So reverse seq, sequence name, sequence, okay, and output file. So now if you can check also, so basically it starts with A, right? So basically A will go at this part and becomes other. Okay, so A becomes T, right, and G becomes C, right, the complement as well as reverse. So they have reversed in their direction and also complemented. Okay, so that just one command and it gave you the reverse, the ORFs or translation or 6 reading frames or the reverse complement, right. Okay, so EMBOSS is one tool. Then there is another one which is a Galaxy platform, which also contains EMBOSS, and it's also very comprehensive web-based platform. Okay, where you can create a session and do your job. It is also free, no installation needed on your local system. Okay, no complex command like we have seen a little bit here, and quick and basically open source, and so many tools. Almost 9,500 tools are available at this site. This is also a site of choice for genomic analysis, right. So like it will open this kind of window, like here you can find all the tool on the left side. In between you will know that what tool you are running, or okay, what you have selected, and this is your past activity, the history, that what all you have done as a user, right, analysis history. Okay, and you can also get data in the Galaxy platform. Either you upload file from system, copy paste, or fetch from the publicly available sources also, which you can get directly by using the standard accession IDs that are there for those sequences. Okay, so it can connect to the publicly available databases like NCBI, UCSC, etc., and get the sequence for analysis. So you don't have to always upload from a system. You can just specify the accession number. It will download from publicly available source and do the job, whatever analysis you want to do based on the I think more than 9,000 of analysis that are possible, tools which are there. Okay, so it gives you history and those kind of things, also utilities where you can know that what activities you did in the past, and also option to download the past files, right. And see, you can see that so many programs are there. Even EMBOSS is also here, NCBI BLAST is there, right, the common ones which you do, seq-tk is there, right, and machine learning applications are also there, many, ChIP-seq and RNA-seq. Okay, assembly annotation pipelines are there, right. So FASTA and FASTQ, genome file manipulations, and SEMBEM file creation, nanopore data analysis, so many things are already available here. Okay, and this is one good platform which you can directly use for your work. Okay, and basically it will tell you that analysis waiting, running, or success or fail. See, basically it is running your analysis on their own system, and so many people might be using worldwide. So if you are giving a big job, it may take some time. So but it is, at least your job will be done, and it is better to, you know, if you don't have required resources at your end or the program to do that, it is better to use a program which is available and wait for some time. Okay, or you can download on your system like EMBOSS toolkit, or write your own scripts and programs and run, right? And all options are fine for analysis, and it also gives you a visual display of the results, okay, which is publication quality, and you can use that for you know, reporting your results and for your figures and images in your publications. Okay, another program that we discussed earlier was Clustal W, which is also available at EBI site and other sites also, and you know, that is both command line and GUI based. I just mentioned that just to refresh the previous thing that we did earlier. So it can provide you an option to you know, upload your program sequence and do the multiple sequence alignment on your system, and also using the GUI based or the online based. So three options are there: standalone command line, okay, standalone GUI based, or publicly available Clustal W, right? Okay, now when we are seeing all this, we should also know that there is so much data these days that your local systems, if you like the normal like Core i5 processor and you know, medium RAM like 8 GB, so it will take years for the analysis to complete, like any metagenomic data or human genome data, right? And you cannot wait for years. So that is why you need very high performance systems. So there are various options. You can have a very strong workstation, okay, one option where all the processors are on board and you know, high RAM is there, okay. So maybe like 48 or 96 processors are there. Each processor can support 2 jobs, so basically, okay, 2 threads, so it can become like 96 becomes a 2 times of that, okay. So now this is one way. And high RAM, like typically 64, 96, or even like 2 TB RAM systems are also available, right, for analysis, which may be needed and are a requirement for heavy duty genome assembly and analysis, right? Okay, or you can always have clusters which have smaller nodes, okay, and connected to each other in a with a very fast you know, network, and your jobs will be divided into smaller nodes, okay, and it will be completed and you return back to you as the output. So HPC, high performance computing, is one of the things that you will come across, and you may be having these kind of facilities, okay. So basically it uses heavy duty workstation or supercomputers, right, to solve advanced problems, and okay, so basically a typical 3 gigahertz processor can do like 3 billion calculation per second, but right now we are talking of like much bigger than that, okay, in HPC, right. So in HPC, basically there are multiple nodes, okay, which are connected to each other, right. So all of these nodes are a separate, or basically you can say a unit containing required amount like CPU and RAM and you know, a little bit of hard disk, right. There could be a data center associated with your HPC, okay, and all these are connected by very fast network and basically communicating with each other, right, and all of those should be of the same specs. If they are of different specs, then basically whichever is the weakest one may slow down your total processing. So ideally all the nodes should be of the same specifications, right, or same software, same OS, right, should be installed on all of those and connected to the data center also to store the outputs, right, and it can do massively parallel calculations with high performance and efficient computing, right. So basically a HPC is comprised of multiple compute servers containing thousands of cores, okay, suitable RAM depending upon the task that you want to do, right. Each of the computing server is called a node, okay, which are connected by the network edges, right, and basically as I mentioned, each component must keep pace with the others, okay. If one is slow and others are fast, basically that slow one will slow down the entire thing, So basically all the components should be appropriately designed, and at least the nodes should be of the same spec, the connectivity between them should be the same, OS should be the same, otherwise one will slow down the others also, right? So like this is one rack, you can see that basically it has one systems, okay, aligned like this, okay, and basically these nodes are, there's a rack of nodes, and this picture is to be taken from internet, and basically you may have also find, if you have HPC in your institute or organization, that these are rooms, big rooms packed with systems, and okay, rack of servers, and inappropriate cooling conditions, because all of these are doing computing and generating a lot of heat. So that heat can become an issue, and many times like it may melt your processors also, that much heat is generated. So basically cooling is one of the important things that also is considered here, okay. That is why you can see that these are clean rooms and basically or cooled up rooms, right? And HPCs are very common these days for advanced computational research, academic or commercial, weather or climate predictions, because it is huge calculation, astronomical calculations, right? Data sciences and data analytics, like you want to analyze the data of stock markets or all the public buying happening at Amazon or Flipkart or these kind of sites, okay, right? So basically those really require like this kind of efficient and parallel systems, right? And there are, like, you can find the list of top 500 servers from here, okay, from this wiki page, which tells you that what is their specification, and that will give you an idea that what kind of systems people are currently using in HPCs. And basically in genomics they are very, very important, like genome assembly, for example, aloe vera genome, which is 16 GB, and human genome is how much, 3 GB, so 2.93, okay. So it is 5 times bigger than the human genome. So you can imagine that if human genome took almost like 20 years for completion, assembly, and like that, aloe vera genome with so much data, 16 GB data, if you want to analyze and assemble and okay, do annotations, you really need such high performance computing systems, okay. Even for alignments, like if I do human and chimpanzee alignment, or aloe vera versus another species of aloe vera genome alignment, right, 16 versus 16 GB, so it requires massively parallel and this kind of heavy duty systems. Dealing with metagenomes, which is basically all the genomes which are present in an environment, okay, analyzed together, so that is also a challenging. AI and ML approaches, in which basically training data or the test data could be very, very large, so you really have to be have these kind of tools, right? Modeling of cells, like how a cell functions, okay, doing all the statistical and theoretical calculations about how the cell, whatever is happening in the cell, basically that may also need these kind of systems. Molecular dynamics simulations in proteins, like you are trying to predict a structure or do some kind of molecular dynamics with the structure, right? So those is also heavy duty calculations and may need these kind of servers, okay. Alignments and basically any complex computing problem, if you want to do, like HPCs will do much faster as compared to like simple standalone PCs, right? And then basically I've been talking about these. So what should be the specification for your system depends upon your task. Sometimes it could be more CPU intensive, some kind it could be more memory intensive, right? Or of course hard disk and storage will always be less, right? Whatever you think of, so you have to think of an appropriate storage, or even backup of backup later, because once the data is deleted it will be lost, okay. So backup is also important here. Connectivity, right? So typically like how many cores should be there, okay, there is no limit, right? But still you can see that what program you are going to use, and you can check that, okay, what other groups have used that program for what kind of data, what was the data size on which they use the program, and what was the time taken and resources for doing that, and based on that you can plan your system, right? There has to be good graphics and display, okay, and some USB ports, of course, for basically connecting various kind of accessory tools, okay. and there could be an option to have processors on board, multiple processors, like you can have a single workstation with 96 processors on board, like 424 CPU processors on a single board, or you can have 4 different nodes with 24 core processor, right? So both are like so if you combine those 4 together, it will become a cluster containing 4 nodes with 24 core processors, or you can have all 4 processors on a single board and still do a quad core processor, okay? Do your job, right? So these are the options and choices that you have to make based on your analysis work and requirements, right? And basically having a combination of these also helps sometimes, like having a good workstation as well as HPC cluster access is important for computational genomics. Okay, so in the today's lecture we looked at basically the various genome and basically genomic analysis packages, right? Online ones such as Galaxy, EMBOSS, okay, EMBOSS, which you can also download, Clustal W also you can download, okay. So many others are also there which we could not touch, but basically they would be similar to what we have described here, right? So basically the way to find that will be read literature and see that what all softwares are available. There are dedicated versions of like nucleic acid research, bioinformatics, these kind of journals where the list of these softwares are published, right? And then standalone versus web-based analysis. So both options we discussed that what is the benefits of these, and we introduced the HPCs and a little bit of specifications that you may need, okay, for your analysis. And so this is good for today, and then we will meet in the next class. Okay, thank you.